Okay, this is where I switch to looking at a cellular alteration. Now this is using slice electrophysiology done by Leslie Whitaker in my lab. And also, in her case, she's looking at food self-administration, where animals are learning to press a lever for 1 day, and then after 10 days of expression, then we call this the day 10 trained animals. And what we're trying to do is, after the very first day, we look at Phos expression. Day 10, we look at Phos expression. What changes occur within the neurons, in this case, the prelimbic cortex? What changes happen going from day 1 to day 10 during learning, rather than just expression afterwards? So when you look at the behavior and look at the active lever pressing, as they gradually learn to press, you'll see on day 1, they're not very effective at learning how to press a lever under these conditions. We purposely did not food deprive them, which usually makes them press a lever and learn more or faster. But by day 2, they're already learning to press that lever for food, and they're quite good at it by day 10. But if we went and perfused the animals after day 1, their first session of training, or after the 10th day of training, you'll see that there's a great deal of Phos after day 1 and day 10, especially when you compare it to animals that were taken right out of the home cage without any kind of training on that particular day. And you see the Phos expression over here. The dotted line indicates the amount of Phos that you get in an animal that was taken straight out of the home cage on day 1. There's not a lot of Phos expression. But when the animal went through 1 day's worth of training, you see a great deal, a large increase in Phos expression in the prelimbic cortex. There's a great deal of activation of those neurons. But if you did it after day 10, there's a decrease in the amount of Phos expression. For some reason, the amount of Phos that's activated from day 1 to day 10 decreases. Now, there's a host of reasons for this. People talk about novelty as going down, but what does it really mean when you talk about novelty? Basically, there's a host of things. But in the beginning, the animal is responding and paying attention to a whole host of cues and stimuli in its environment. It doesn't know ahead of time. You didn't explain the experiment to them. So it doesn't know ahead of time which are the relevant stimuli. And we like to think of it that there's a refinement of the ensemble that goes along with the refinement of how the animal responds to the stimuli in its environment. Now this shows up, this pattern of Phos expression, day 1 to day 10, shows up in a whole number of other brain areas. Now, just for the sake of showing you these neurons are at least reasonably important for behavior, we performed a DONO2 inactivation experiment. They were trained from day 1 through day 9 for that food self-administration, 1 hour each day. But on the 10th day, they are trained again, but on that day, 90 minutes, and this is, sorry, I should have said, these are the phospholaxi transgenic rats. On that day, basically you're inducing beta-galactosidase in those Phos-expressing neurons, and then you're trying to kill off those neurons by injecting that DONO2 drug, that DONO2 prodrug. And then testing, if you killed off those neurons, you should decrease behavior on test day. And over here, you look over here, this is going from the vehicle injection to the DONO2 injection into the prelimbic cortex. There's a significant decrease in lever pressing, indicating the importance of these particular neurons in that behavior. You might notice in a lot of these DONO2 experiments, it never makes it go from 100% down to 0%. One of the reasons is that one brain area is not responsible for the entire behavior, is the way I like to look at it. The whole brain is responsible for the behavior. But for the sake of this experiment, it's showing you that these particular neurons are important for mediating that behavior. This is where we bring in the other transgenic animal called a Phos GFP rat. It uses the CPhos promoter, and it encodes the green fluorescent protein, which allows you to detect these Phos-expressing neurons in a slice preparation. And they'll make that green fluorescent protein, which you can see. These are very thick slices, so they're not going to be super pretty. But it's enough to allow you to see the Phos and GFP expressing neuron, so that you can then switch to differential interference contrast using infrared. What this does, it allows you to patch onto the cell and record its electrophysiological characteristics, just to put a hole on it. And what basically Leslie does, she records from these particular neurons, the positive neurons, and compares it to the activity of a nearby neuron, the Phos negative neuron, and see what's the difference in the responses to day 1 and day 10 training in the Phos positive and Phos negative neurons. So there's something we quite often look at, things called synaptic alterations, which are alterations in synapses that land on particular neurons. But in this case, we're looking at what's called intrinsic excitability. It's just basically the set of characteristics that tell us that they're involved in making a cell less responsive or more responsive to their inputs. In this case, out of many different things Leslie looked at, one of the things you inject a certain amount of current and you see how many spikes, how many, in this case, like action potentials you can induce with that amount of current. Now, if you look at the majority of neurons that are phospho-GFP negative, when you look at the day 1 neurons, there's a certain number of spikes. But when you look at neurons from animals that were taken after their 10th day of self-administration training, there's a decrease in the amount of spiking for that same amount of current. They decreased their excitability. And this is shown in a graph over here. Day 1 is in the solid circles, and then day 10 is in the open circles. There's a decrease in their ability to be excited for a given amount of current. Whereas in that small number of neurons that were strongly excited, the Phos positive neurons, their amount of spike response to that same amount of current actually increases. And this is shown over here. The number of spikes on day 1 is shown in closed circles, and then day 10, the open circles, there's an increase in the number of responses to that same input. There's an increase in excitability. They're going opposite directions. So this is not some generalized effect that you get in all neurons. So why would this happen? She checked dozens and dozens of different electrophysiological parameters. One of them is input resistance. And basically, what she had found is that going in the Phos-GFP positive neurons, that small number of neurons, that going from day 1 to day 10, there's an increase in input resistance. Now, this is easy to explain. Basically, if you have increased resistance and you increase the amount of current that you're putting into a neuron, that makes the voltage increase for that particular cell more responsive, and that induces more spiking. So she felt like she had part of the story as to why the Phos positive neurons would increase their excitability. And there's a lot of work to determine what's responsible for that. On the other hand, the vast majority of neurons that were not strong enough activated to make Phos, the Phos-GFP negative neurons, she looked at a lot of things, but one of them is something called the medium after hyperpolarization. After basically after the action potential, you get an after hyperpolarization. In this case, this particular type is called medium after hyperpolarization. And it's part of it effectively keeps the cell less responsive for a longer amount of time if you increase the amount of, or the degree of medium after hyperpolarization. And basically, when you look at the Phos-GFP negative neurons, you see going from day 1 to day 10, there's an increase in this medium after hyperpolarization, whereas this did not happen in the Phos-GFP positive neurons. So there's different mechanisms being operated on during learning in the Phos-GFP negative versus the Phos-GFP positive neurons. And fortunately, this is one of a few things that are easy enough to do. One of the things that mediates this medium after hyperpolarization are RSK channels, well known for quite some time. And one of the drugs that will inhibit that, or chemicals, is apamine. And apamine will block those RSK channels. And she had shown that using apamine on the day 1 trained animals had very little effect, whereas using apamine on slices from animals taken after their 10th day of training, apamine had a very significant effect. You can see it quantified here going from day 1 to day 10, showing you the effect of apamine on this medium after hyperpolarization. Now, going back to the original finding, that excitability change that she had seen, when you look in Phos-GFP negative neurons, going from day 1 to day 10 in the absence of apamine, you see that decrease in excitability that we talked about before. But if you have apamine on board going from day 1 to day 10, basically, apamine has the ability to block that decrease in excitability. So this is quantified here. Apamine has very little effect after day 1 of training and has a strong effect after the 10th day of training. So just summarizing here, you have some effects in the phospho-GFP positive neurons that increase excitability due to input resistance, and in the vast majority of neurons that are phospho-GFP negative, you get decreased excitability, increased medium after hyperpolarization, and increased amount of what SK channel contribution to that. So I'm going to wrap that particular part up and just say what I've been talking about is the food self-administration, looking at food as a reward, looking at the rat prelimbic cortex. But we had seen previously what we called synaptic alterations too, which quite often you see a lot of that in the electrophysiology papers. And they're very unique changes that are happening only in the Phos-positive neurons. In this case, most of the cases we get an increase in what are called silent synapses. It's an increase in the number of neurons that don't have a particular glutamate receptor, the AMPA receptor type. They have NMDA receptors. They do not have the AMPA receptor type. And this happens uniquely in the Phos-expressing neurons and not the majority of neurons that are Phos negative. So what we see is a set of unique alterations that are happening only in Phos-expressing neurons, only those that were selected by those cues in the context, and that we have seen are actually mediating that particular behavior. So this brings us to a definition, a terminology issue. You might have seen the term engram used a lot. I mean, it's a lot of people are using that, especially Tanagawa, who's taken a lot of credit for a lot of stuff. He uses the term a lot. And when you go back to what the history of the term, it basically means what that trace, that physical trace that's left behind after training that allows that long-term memory to be stored. Being able to manipulate these cells using optogenetic techniques or what we call the DREAD techniques, it's not enough to show that the neurons are active. They could just be relaying the signal. It has to be more than that. You have to show that there's some physical trace within these particular neurons that could be mediating that long-term memory. So we've been starting to look at these kinds of changes, these long-term physical changes in neurons that seem to mediate the behavior. So we think we're on the right track. The thing that we've been working on for a while, unfortunately, transgenic rats, especially when you make double transgenic rats, it can take years to get the system working. And the system works. What we're trying to do here is be able to manipulate all these molecular and cellular changes only within the Phos-expressing neurons. And the system does work, as I was explaining to Sega, but only a current system only works in about 50% of the animals. Why? I wish I knew. But so I'm not going to get into it. It's not ready for prime time yet. But basically, it's a CFOS promoter, drives expression of the Cre recombinase. But we don't want this promoter to be turned on all the time. We don't want it to make this Cre recombinase molecule, because that Cre recombinase molecule, once it's turned on, it initiates a process that is permanent in this case. So we have a second gene, that's the TET repressor, that binds to the TET operator on this part, on the promoter, and keeps it silent. That's the other word I learned in Hebrew, sheket. So yeah, it's used a lot around Tel Aviv, I found out. I wish somebody had used it. There's another thing I find in Tel Aviv too, is if a taxi driver stops at a corner, you know he's going to be gone in about 15 seconds anyways, but everybody hits the horn and keeps on going. I wish I said sheket at that time, but anyways, so when you add tetracycline to these particular animals, and we've done it many times, it releases the repression of that promoter and allows the CFOS promoter to be turned on and Cre recombinase in those activated neurons. And it does turn on different kinds of Cre-dependent genes. like the optogenetic proteins, in our case, heterodopsin or other fluorescent proteins. And we use this to tag the cells in a permanent sense. But like I said, I'm not going to go into it because it's not ready for prime time. All I can say is, following the tetracycline injection, there's about a 6-hour window where anything that's activated within a span of time basically gets permanently labeled. And it's much shorter than the other 1, 2, or even 4-day windows that people have had before. But I wished I could have talked about more about this. But basically, I'm just wrapping up. So unique alterations in activated neuronal ensembles may mediate that long-term memory. What I'd like to be able to do, and I mentioned that, is look at, to test the causal role. Are the molecular and cellular changes that we're observing actually mediating that particular behavior? And if we can show that, this would be the physical trace within neurons that we show are mediating the behavior that we could call the engram. And that's what I'd like to be able to show, not just for food self-administration, for drug self-administration, but also for food self-administration and a host of other ways. So I think by this point in time, we have procedures to truly look at the true mechanisms of learning and memory in animals that are behaving, and look at them in the neurons that are actually mediating the behavior. And with this, hopefully I'm on time with everything, I'd like to thank the host of people who worked with me on developing this stuff. I'd like to thank Asa K. Koya was critical in the beginning for electrophysiology and the Dono-2, an activation procedure, working with me on that. Daniel Gesbarber on the facts, and Leslie Whitaker for the electrophysiology. And Brandon Warren, I'd shown you his work with self-administration and extinction. Javier Rubio has really followed up on using facts, not just for mRNA changes, but he found you could use it with frozen tissue, so we're actually going to be using it on human brains too, to be able to separate cell types and perhaps Phos, from human brains. And it actually works better with frozen tissue, which blew me away. And on top of that, we're using you could use antibodies against proteins that are of interest and measure the amount of proteins as they're flying through the facts, and also protein phosphorylation. The other reason why it's good to use frozen tissue, because you need to do that when you're looking at phosphorylation. So, and my colleagues, Yavin Shaham, definitely, a lot of, he and I worked together on most everything. And then Roy Wise, who was basically channeling Donald Hebb, although I didn't know what the heck he was talking about for the first 3 or 4 years. But you sit down with him for like these 2-hour conversations. You just have to listen, But he was basically channeling Donald Hebb, who was one of his instructors back a long time ago. And with that, I'd also like to rapidly mention Tarun. He's actually a biomedical engineer who's I'm looking forward to working with a lot. There's another technique I'm trying to get is getting these 1-micron LEDs inside that have certain ID optical detection, how I should, optical ID mechanisms, so we can actually measure neuronal activity in a great number of neurons to look at ensembles more accurately. And with that, I'd just like to thank you for listening. And
I wonder why specific behavior would recruit specific neurons when the learning occurs, why those neurons would express Phos and not any other neurons in the population. I remember there was a notion that those neurons are easier to be activated with a lower threshold, but you showed me that actually is the same.
yeah, what selects them are the inputs. To me, it's just how they respond to the cues and the context. Okay, we'll select which particular neuron inputs from the other afferent brain areas are being turned on. And that specific pattern of inputs will integrate onto the neurons in any receiving area. It's their excitatory input that selects them. And I don't think it has to do, and we've shown, as I showed, that changing the cues of the context will make the other neurons be Phos expressing, as you mentioned. The thing I did not mention, and I kept telling myself, there's this critic, this guy talking to me all the time, and it's saying, mention the cellular phenotype issue. we always test which of these neurons are Phos expressing. If we're looking in the cortex, the vast 90% of them are glutamatergic, 10% are GABAergic. If we're looking in the medium striatum, the vast majority are medium spiny neurons, equal D1 and D2 neurons, and low amounts of all of the different kinds of interneurons. They basically, you're reactivating almost the same proportion of cellular phenotypes that you would find anatomically, what you would expect. The only cell type that's overrepresented are the parvalbumin neurons. The rest of them are, it's not just one type of cell. That's basically what I'm getting back to. Some people would say, oh, it's just the D1 neurons, but it's not like that. Does that answer the question? Or?
But I guess many neurons, much more than 1% in that brain area receive this stimuli, right? Only those 1%.
Only, yeah. Well, that's what I was trying to get to, basically. They are receiving what I call most neurons receiving, being activated to some extent. But basically, I think what it is they are receiving the most persistent excitatory input, the strongest and the most persistent. Basically, I think of it as overwhelming the ability of the cell to buffer that calcium that's coming in, because most cells will buffer that calcium rapidly. But if you persistently activate those neurons strongly enough, it'll overwhelm that calcium buffering capacity and then activate the system that turns on Phos, and I think that's MAP kinase. That's what I think is the mechanism for selecting only the tip of the iceberg, just the most strongly, most persistently activated neurons.
Were you able to compare different ensembles for different rewards and see it's really different, not just a general reward ensemble
That is actually what Javier Rubio is doing right now. And basically comparing the heroin versus the cocaine. And it's, unfortunately, we would have had the data already. But he's originally from Chile, and it took like 6 months to get the immigration papers going. So
it will be easier now.
Yeah, oh yeah. That's right. I don't know how they feel about people from South America versus Mexico there in that administration. But basically, that's exactly what we want to know. It's a critical one to know, because you should respond differently to different rewards. And we'll see.
You said it's context-dependent, the learning. What does the brain perceive as a different context? What stimuli would define context?
Well, yeah. The way I look at it is stimuli that are, as I said, it's a complex set of stimuli, and it's hard to know specifically what in that environment they're responding to. It's the whole set of stimuli, the whole set of cues. You might call it like a configural cue. And basically something that is present during the whole session, and that if they press a lever, it's not dependent on pressing the lever. That's a separate kind of cue. Okay. Because basically the effect of the context can change how those discrete cues, the lever paired cues are, the effects of the lever paired cues can be altered by that context. And you can go into, I'm in front of a bunch of psychology people. You should tell me this. But okay, first year, that's okay. You learn a lot in the first year. But basically, it's something that's present there the whole time and is not directly paired with whatever operant response you're making. Okay? And basically, you would respond to the same question very differently if you were in a different context, like in front of your professors, as opposed to how you would respond if you were around your friends. And that would be the context. You would respond very differently.
Thanks for the wonderful talk.
Okay.
I was wondering, on the non-active cells, there was a specific change in the profile of the channels that are being expressed. What do you think is the mechanism that would actually refer the non-active cells to change this process? Is this a maintenance process that is going on? Yeah,
because this is I knew somebody was going to ask this, but no, it's true. It's a good one. Because you end up with this idea that the Phos negative neurons are decreasing their excitability, but this happens in all neurons, and they're going to be useful for other memories. It's not like you only have one particular memory. So in many cases, many of our experiments have a lot of artifacts of being kept in the home cage situation. What happens if they have multiple memories, like out in the wild? My guess is many of those phospho-GFP negative neurons that decrease, the SK channel immediately after hyperpolarization, probably has happened already in many animals. And it's because they've responded to so many different kinds of cues and stimuli, that variety in the environment. So basically, it's only those Phos positive changes that we're seeing in the selected neurons that are the ones that are relevant, that I think you would find in a wild type animal out in the environment. That didn't answer it. Go back. Could you repeat that again? Sorry, I didn't.
Well, again, so you see a change of the profile of the SK channels.
Right.
So what would keep them, what would make them go at the lower level in the non-active cells? What kind of cellular mechanism would select them?
I honestly don't know. That's the only way to respond to that one. Okay. Oh, okay.
What about, what is the role of the neuromodulators that are related to the rewards, such as dopamine and Phos expression?
Ah, yes. It should be known that dopamine physiologically doesn't do what a lot of people say it does. I mean, dopamine by itself will inhibit neurons. You know, I mean, people have shown that many times. Glutamate is the main thing driving the excitation of most of those neurons. And dopamine will basically, the idea is that dopamine enhances the responses to strong ongoing glutamatergic excitation. And that if there's not a threshold level of excitation, that dopamine will tend to inhibit those neurons. So basically, you have some neurons that are getting some activity, some neurons that are not getting enough activity, then dopamine will enhance that signal-to-noise ratio. I'm a fan of that idea. That's what I think those other modulators are doing. They're changing how you respond to, say, glutamatergic inputs. And for dopamine, I'm sure all the other neuromodulators are probably affecting it in that way.
So you expect that if you, for example, block the dopamine, you will see much more noise in the expression level of the Phos?
Well, yeah, I really, yeah, if you block it, yeah, you probably get, I haven't done it, but yes, you do decrease the amount of Phos. Having said that, we've done an experiment where if we decrease activity using anesthetic and you have the same amount of cocaine-induced dopamine, for instance, you don't get any Phos expression. Whereas if you added back the glutamatergic inputs to, say, the striatum, then you get the Phos expression, and then you get the activity, showing that dopamine by itself isn't driving the neural activity in that case. So basically, I think it's, in most cases, it's that glutamatergic excitatory input that's driving Phos expression, driving behavior. And the other ones are shaping and modulating that particular input.
Yeah.
Sorry. And then,
You talked about ensembles of neurons as encoding for memories. So theoretically, if we take an ensemble and we kill off one or two neurons, the memory is no longer valid?
Valid, or it's no longer present? Yeah.
Encoding now, right?
As far as that particular brain area that you've killed off is concerned, yes, I think that memory is gone in a permanent sense. That information is gone. It's like whenever you get rid of information, you can't just grow it back. Yeah, you kill that particular network. You can, we haven't done the proper experiment, but we have done where we've retrained the animal. And it was just one of those, the rats were there and we decided to retrain the animals. And they don't learn for, say, the first couple of days. It looks like you erased that memory permanently. But what I think is happening eventually is that the axons bringing that information to a given brain area, they're not killed off. There's still enough trophic factors to keep them alive. They're just going to reattach basically to neurons that remain.
Make a bypass.
A bypass, or just select different neurons. Now those different neurons are going to be activated by that same set of cues and context. I think this is probably what happens in other things, like when a cell dies and your brain just due to aging, or eventually you're 80 and you've got a lot less neurons, but you still remember a lot of stuff. And so that's what I think can happen. So, oh, sorry, go ahead. Yeah.
I'm thinking about humans now, like theoretically speaking.
Right.
This ensemble represents the stimulus and the context.
Basically everything, yeah.
And if you, let's say cocaine with people, you want to narrow it down to just the powder. And if you kill off those cells, you kill off the networks. So can you use theoretically different genes for different contexts and then get just the DON2, whatever?
Yeah, you could definitely do it with DON02, and I used to think that was crazy, but there's a guy who does lots of gene therapy trials in the US, Howard Fedorov, and he thought, no, this is the perfect place to do it, where you could insert a transgene that would make beta-galactosidase and use DON02. So that's theoretically possible. But since then, I've seen a lot of experiments that just mystified me, where a person gets a specific killing off of one memory and not the other memory, when what they injected into a brain area of a rat, for instance, was something that should have stopped, say, CAM kinase 2 activity in all of those neurons. But what I'm coming down to the conclusion of is that it's not a matter of just decreasing neural activity. There's all these things that I'm showing you that only happen in these strongly activated neurons. There's a whole sequelae. There's a whole set of mechanisms that are uniquely activated within them. What happens if you interfere with any one of them at that stage? There are certain receptors, for instance, NMDA channels or calcium channels, they're in a specific state when they're activated, that they'll be more responsive to ketamine for NMDA, or certain conotoxins bind to activated open channels of calcium channels. But what about all that stuff inside, the CAM kinase 2, the MAP kinase, all that stuff that's activated in those neurons? If you interfere with that process when the neuron's being activated, then you can interfere basically with the formation or reconsolidation of those particular neurons. And I think in the end, I think what we might be looking at is very similar to the neurons that are selectively being activated during reconsolidation and then being interfered with. So I think we can actually identify those neurons and study those neurons, the effects of reconsolidation using those neurons. Anyways, the reason I'm getting back to that is there's a host of drugs. If that's the case, there are a host of drugs that should interfere only with activated neurons. We're all looking at drugs that only activate certain types of receptors or certain cell types. There has to be a whole set of drugs, I think a whole new pharmacology basically, who only interfere with activated neurons versus non-activated neurons. And I think we've got a whole host of different kinds of things we could look at if we're trying to affect just the ensembles, because the only thing that defines them is their activation state in a given situation. And that we can, there's probably a whole host of things that we could use with humans.
So my last question is about individual differences. Because until now we had this so the hippocampus is responsible for the context-related memories and the amygdala for the fear-related memories and so on. Now we are talking about within these regions we have ensembles of neurons. Are these the same ensembles for me and you, for example? Or
in the most general sense, I believe so. But this is another thing. I love this question because then you get in. I won't go on too long because I could go on for hours about this one. But I'm fascinated by this because what allows an ensemble to form could be due to a whole host of things. Each of you and me, we all probably have different numbers of glutamate receptors that are in our synapses, different abilities to, different excitability for that matter, of neurons once they take this integrated input. Some of us may have, in response to the same memories or a set of stimuli, we might have 0.5% of the neurons being activated in a given brain area. Somebody else might have 2% of the neurons being activated. What does that mean for your ability to, A, say, discriminate between memories, or sometimes having one memory interfere with the other memory? Or is it really interfering? Or is it part of what allowed Donald Hebb to form what I almost call intuition, is the ability of separate kinds of stimuli or separate ideas to interact with each other. People where you don't get, say, a large enough ensembles, they may not get these types of interactions. Anyways, I could go on and on about that, but I think there's a whole set of interesting personality issues and ability to learn and maintain ideas and synthesize ideas. I think there's just tons of interesting ideas we could look at when you're looking for it. We just have to stop looking at just the molecular or cellular changes. And these are the kinds of things I think would be really cool.
Thank you very much.